all of you in this room, and I think actually every intelligent 6-year-old knows that there is a record of life incorporated into sedimentary rocks, the fossil record. And we learned early that some time ago there were dinosaurs. They were preceded by earlier tetrapod vertebrates that were preceded by fish. And really intelligent 6-year-olds know that there's also a record of marine life, such as trilobites, and that this entire conventional record of tracks, trails, bones, skeletons, takes up what's called the Phanerozoic Era, or the last 541 million years. Now, if you look on the right, the Phanerozoic Era is placed in the context of the geologic, the age of the Earth as a whole. And we've heard a lot at this meeting about things that go on in the Hadean and Archean eras. That somehow must connect up to the things that people like Simon Conway Morris talked about, which have their roots in Phanerozoic evolution. And what connects those is the Proterozoic era and its record. Now, there are two reasons why we might be interested in the Proterozoic. One is for its own phenomenology. This is an era nearly half of all recorded Earth history. It begins, or when it begins, the Earth basically has no oxygen in the atmosphere and has a biota made up of bacteria and archaea. And by the time it ends, we have a relatively oxygen-rich atmosphere that supports a biota that includes animals, macroscopic seaweeds, and the like. Between those, both at the beginning and again toward the end of the Proterozoic era, are, as we heard yesterday, ice ages of a magnitude that is unknown over the last 500 million years. Also, the emergence of eukaryotes as a major part of ecosystems. So it's a rich record that I think one can look at for its own sake. And at the same time, it is a record that is much richer, much more complete than the rather fragmentary record of the Archean, and therefore it can actually provide something of a template for thinking about how we might interpret the earlier record, not to mention guide us in the exploration of sedimentary rocks on other planets. Now, if the Phanerozoic, by definition and etymology, is the record, is the time of visible animals, if we place that in the context of a molecular phylogeny, and there's simply a notional phylogeny there, and I understand that just about every note on this will be controversial to someone, but I think everyone would agree that the animals that make up that conventional record of the Phanerozoic constitute only the distal tips of one branch of the tree, and that the deeper record, and therefore the life that we might infer prior to the Phanerozoic era, is largely microbial. So let's look at some sedimentary rocks from the Proterozoic eon. These are some rocks that when I was younger and fitter I worked on for a number of years. These are carbonate rocks exposed in the Arctic island of Spitsbergen. And what you're looking at there is about 1,000 meters of carbonate rocks deposited between about 800 and 740 million years ago. There are no skeletons, no bones, no tracks or trails, no evidence of animal life at all. And so at this point, the search for life becomes a practical one. If we believe that the antecedent record was microbial, should we expect the sedimentary rock record to preserve a tractable record of tiny microorganisms? This also illustrates one of the advantages of essentially working out our rules of the game for exploring deep Earth history, that if you look at this pile of carbonates, if you would take all of the early Archean carbonates that have ever been measured from basically everywhere in the world and stack them on top of each other, they would be about as thick as the little beige band right on top of the black one at the corner of that picture. So the record is indeed much richer, and it is preserved rather differently. So what do we see? If we hone in on just one bed of those carbonates, you can see here that there are these wavy laminated dolomites. We know from just comparisons with the modern world, where we can see processes in action, that the kind of wavy textures that you see in that outcrop are generally associated with places where microbial mats interact with accumulating fine-grained carbonate sediments. That tends to happen in coastal environments, and we can see it in places like the Bahamas today. If you look in the upper center of that picture, you'll see something that's bowed up. In the button-down parlance of geologists, that's called a teepee structure, and they tend to form in the supertidal environment, that is the most landward and most frequently exposed of coastal environments, where evaporation causes precipitation of carbonate minerals, and the pressure from that precipitation within pore waters causes beds to buckle. So what we learn from this is, first and foremost, is that not only does the stacking of sedimentary rocks give us a record of time, but physical and chemical features in those sedimentary rocks tell us something about environment. So we actually have a time and space framework for thinking about life. Now, if you took a piece of that carbonate and just made a thin section, put it under a microscope, all you would see was interlocking crystals of dolomite, not particularly interesting to the paleontological eye. But if you went to those black concretions that you see in there, those consist of silica, SiO2 or chert, which formed soon after the deposition of the parent carbonates and actually grew within the sediments at the expense of carbonates in a way that actually preserves micron scale textures of the original sediment. So if you take those cherts, make thin sections, put them under the microscope, what you will see is what you see in the bottom, which are well morphologically preserved microfossils. Each cell-like unit there is about 4 microns in diameter. So the good news is that we actually have mechanisms in the Proterozoic eon that preserve in an identifiable and tractable state a microfossil record. And indeed, when you look across different environments in this package of sedimentary rocks from Spitsbergen, not only in silicified carbonates, but in fine-grained siliciclastic rocks, that is to say shales, we find a variety of things that on the right-hand side are very similar to living cyanobacteria, and on the left-hand side, we see things such as on the left, where we have a fragment of a macroscopic structure that's synocytic in organization, very likely green algal. We have 100-micron tests, very similar to those of different kinds of testate amoebae. We find cysts like we see in the lower center that are about 500 microns in diameter. So there is a microfossil record. A more conspicuous record turns out to be the record of stromatolites that most of you are familiar with. The lower left is sort of the mecca for Precambrian paleontology. That's Shark Bay in Western Australia, where again in the modern world we can actually see the processes by which microbial mat communities interact with sediments to produce these upwardly doming laminated structures. And on the right you can see stromatolites that were nucleated on just blocks, exactly the way the Shark Bay stromatolites are, and then accreted by the trapping, binding, and precipitation of sediments associated with microbial mats. And as you see in the cliff behind that skier there, you sometimes see structures that are several meters high and tens of meters thick, which are actually bioherms or reefs, which are constructed by these microbial communities. So again, there's a macroscopic, conspicuous, and widespread record of benthic microbial communities preserved in these stromatolites. And then, of course, as we've heard a little bit about earlier in the meeting, and most of you know, there are at least several chemical ways in which life can impart a fingerprint to sedimentary rocks. There is an isotopic fractionation associated with autotrophic carbon fixation, so that if you look at, in the Bahamas today, the carbon isotopic composition of carbonates and compare that with the carbon isotopic composition of biologically produced organic matter that is sedimented with those carbonates, they will differ by about 25 parts per thousand or per mil. And broadly speaking, that kind of fractionation pattern between carbonates and organic matter really characterizes the known sedimentary record. So we can start to interrogate the microbiological carbon cycle in the Proterozoic. One can do the same thing for sulfur as well. And indeed, under certain conditions, there are molecular fossils preserved. DNA and RNA are too good to eat, so they basically are not found in rocks of this age, but lipids can actually preserve. So we have things like steranes, the geologically stable forms of sterols, hopanoids derived from bacteria, and independently of the morphological record, these can enhance our sense of microbial physiology and diversities in the Proterozoic. So in rocks that are 750 million years old, we see a fairly extensive record associated with sedimentary rocks that have textures that in general we understand from looking at modern analogs. If we just for a moment double the age and go back to things that are 1,500 million years old, these are some rocks in the upper left from northern Siberia, and basically everything I just said about 750 million year old rocks holds for these. There are microfossils, those are 10 micron bars, some of which are comparable to cyanobacteria, others of which appear to be eukaryotic. There are carbon and sulfur isotopic signatures, there are molecular biomarkers, and there are stromatolites. I like this picture in the lower left for two reasons. One is that my field partner here, a man named Misha Semikhatov, is precisely 2 meters tall when he has his hat on. So he's a great guy to have in the field so that you can get him as a scale for any picture that you make. And if you look at that picture, you might say to yourself, oh, I see that little bun in the center of the picture is a microbial bioherm. And you're exactly right. That's a little reef, maybe twice the size of this lectern. But if you actually look at the surface on which Misha is standing, it is a domed surface, and it is part of a stromatolitic bioherm that's probably about the size of this room. And in fact, the entire wall of rock behind Misha is part of a still larger bioherm that would be somewhat larger than this building. So again, we see that through the Proterozoic eon, microbial mat communities are largely surfacing environments from the coastline all the way to the bottom of the photic zone, and they can accrete structures that have dimensions equivalent to those of modern coral reefs. Now, I'm not going to talk about the Archean. We've heard a lot about it, except to say that this well-preserved record of life in Proterozoic rocks does provide us with something of a baseline that can help us to interpret the deeper record. So what you see here on the left are stromatolites that have been described by Abby Alwood and others from the Pilbara region in Western Australia, around 3.4 billion years old. And if you look at them, the bottom half of the left-hand panel is very comforting. You see these domed laminated structures that look much like the stromatolites we've just seen, but it becomes a little more disconcerting when you continue upward and notice that the fabrics now become vertical. What you're looking at are actually precipitated acicular aragonite crystals that precipitated on the seafloor. This is an environment where carbonate was precipitating in remarkable fashion, and we know from both today and Proterozoic examples that when you have these high rates of carbonate precipitation, you can make domal laminated structures that are not templated by microbial mats. So simply finding something that's domal when the textures are precipitated is probably insufficient to declare victory. And indeed, when you look at the lower right-hand panel, I mean, there's nothing that seems particularly biological about that. It's just a stacked set of precipitated carbonates. On the other hand, by looking at Proterozoic examples, we find that if you go in and look microscopically at petrological textures, you can sometimes find fingerprints of microbial mats. And indeed, when Abby and I and others looked at these in these examples from the Pilbara, we do see petrological evidence that there was at least a microbial influence on these. But you really need to get at that level, and that's a lesson from the Proterozoic. Houman et al. Have done similar things in rocks of comparable age from South Africa. And indeed, there's another thing that we can learn from the Proterozoic that can help us think about microstructures in the Archean. Certainly, the search image for exploration for microfossils in the Archean comes from the success of finding microfossils incorporated in chert in Proterozoic rocks. On the other hand, one thing we know from petrology and geochemistry is that many early Archean cherts have been influenced by hydrothermal fluid flow. And so we might want to ask, is there an influence on the preserved textures of those rocks from hydrothermal activity? And a way to evaluate that is to go into the Proterozoic, where in general we have well-preserved fabrics and fossils, and then look at local hydrothermal fluid flow and see what that does to the textures. So what you're looking at in the lower right-hand corner is a thin section of silicified carbonate from some 740 million year old rocks in Canada. If you look at the brown areas on the left and right, they have textures which are very similar to those of early diagenetic cherts in the Proterozoic in general. But the white and purple areas that you see through the center of the slide are areas of concentrated hydrothermal fluid flow. And within those, all of the original textures of the cherts have been obliterated, but as you see in the two panels above that thin section, that you both have the mobilization of organic matter and then its recondensation, forming 10 to 20 to 30 micron spheroidal microstructures. Now we know that there's fluid flow in the Archean. The upper panel on the right shows organic matter condensing around a rhombohedral crystal. And so we know that happens. So the question is, can we then make a judgment about whether what we're looking at in these very old rocks is associated with hydrothermal activity or maybe, excitingly, true biological remains? And again, I'm not saying that all things that have been reported as fossils in the Archean are other than that. It's just that there's probably one more loop we need to go through to have confidence. And this is where that preservation of textures comes. The expectation, at least for the work that we and others have done on hydrothermal alteration in the Proterozoic, is that it will tend to obliterate textures. So if on the left, where we're looking at rocks that are a little more than 3 billion years old from South Africa, if you have preserved textures on a coarser scale, and what you're looking at there is simply a shard of some laminated, possibly matte material, that actually contains 8 to 10 micron spheroids. Again, I don't know that these are unequivocally fossils, but at least they are in the context of preserved texture. So again, I think that the experience with better preserved materials in the Proterozoic can at least help us as we evaluate this precious but limited record of the Archean. Now, just a few words about Cyanobacteria in the Proterozoic. Many of the Cyanobacteria-like fossils found in Proterozoic rocks seem to be generally similar to living cyanobacteria. There are some populations that are very specifically similar. So if you went to the Bahamas today and looked at ooids, these little concentric carbonate balls that accumulate in the tidal zone, you would find that, as we see in the lower right there, they are festooned with endolithic or boring cyanobacteria that basically burrow into them and live beneath the surface of the ooid. They are diverse. They have, for cyanobacteria, rather complex developmental patterns. They show evidence of behavior in the sense that today we can see things grow down into the ooid until they reach a point of light limitation, and then they grow back up again. We see the same thing in the Proterozoic. What we're looking at in the upper left is simply cross-bedded ooids there's a silicified ooid in the lower left, and you can see that all around its surface it has these endolithic structures, one of which about 1 millimeter long from end to end is seen in the upper right. And in this case, we actually have several populations that are essentially indistinguishable from modern populations found in ooids today. And so that we think this gives us a rare calibration point for looking at molecular clocks, for example, in cyanobacteria. And then there are these things in the upper right. One thing that we find in rocks beginning around 1600 million years ago are these sort of elongate cigar-like structures. That would be about 60 microns long. They are very similar to the aconites of Nosticalian cyanobacteria, which some of you will know is the one clade of cyanobacteria that differentiates different cell types. It has been suggested that maybe these are something else, but one of the things that I think helps us to interpret these structures as aconites, therefore again providing a calibration point for cyanobacterial evolution, is that they are uniquely associated with very short cellular trichomes whose dimensions are precisely those of these aconite-like structures that they're found among. In the modern day, when aconites germinate, they form things called germlings, which are very short trichomes, indistinguishable from these. So again, I think this tells us that by 1600 million years ago, we have gotten to the point of cyanobacterial diversification where we have those cyanobacteria that differentiate cell types. There have been a number of cyanobacterial phylogenies published over the years. This is just one from Betty Schurmeister et al. What this suggests is that the Proterozoic was a time when cyanobacterial diversity was unfolding, and indeed I think that's very similar to the story we get from the fossil record in general. Moving then to eukaryotes, again, as all of you know, eukaryotes are both genetically and cytostructurally different from prokaryotes. And so the question is, can we actually document a record of eukaryotic evolution back through the Proterozoic to complement that of bacteria? And again, the first question is practical. That is, when we find a microfossil, how can we tell whether it's eukaryotic? It doesn't have a membrane-bounded nucleus. It doesn't have multiple linear chromosomes. It doesn't have small subunit ribosomal RNA or any of these things. All that preserves, for the most part, are walls. So if you're a eukaryote that didn't make a cell wall, there's a fair chance your only possibility of entering the geologic record would be as a molecular sterane microfossil. So here's one example, something called Shuyu from 14 to 1600 million year old rocks in China. It's large, about 150 microns. While that might prejudice you toward a eukaryotic interpretation, it's not by itself definitive. On the other hand, these are festooned with regularly distributed processes. And if you look at the cell wall, particularly in the SEM in the lower right, it basically consists of these tessellated plates. And when you look at it in TEM, work done by Emmanuel Javot here, you can see a complexity of wall structure which is very similar to the complexity we see in eukaryotic cells, very different from the ultrastructure that we would see in, say, cyanobacterial envelopes. So I think together, size, wall complexity, and morphology tell us that this was a eukaryotic cell. And just to quote Tom Cavalier-Smith, a prominent protostologist, it says, cysts with spines or reticulate surface sculpturing would probably have required both an endomembrane system and a cytoskeleton, the most fundamental features of the eukaryotic cell. And I think that's right, and that gives us confidence that, A, that we're looking at a eukaryote, and also that preservable features of eukaryotes can actually allow us to make some inferences about features of cell biology that are not well preserved. And again, that particular fossil is over 1,400 million years old. Now, I'm going to skip through the next thing because I already hear the hoofbeats of the end of time coming up. I did want to say that with Emmanuel, we've just published a paper on some 14 to 1500 million year old rocks from northern Australia, and these have a moderate diversity of things that we think are eukaryotic. They are not easy to place phylogenetically, but because of some of the forms, we think we can identify some of them as being osmotrophic, for example, as opposed to photosynthetic. We know that we can see evidence of cell differentiation. I think people sometimes think that cell differentiation is the special province of multicellular organisms, but most eukaryotic organisms differentiate different cells during their life cycle, and we do see cysts. I'm going to just zip through that. We start seeing, through the work of Nick Butterfield, things that we can actually identify phylogenetically by about 1,050 million years ago. These, I think, are well interpreted as red algae, which tell us that not only have crown groups come into existence within the eukaryotes at this time, but photosynthesis has come to them, simple multicellularity has come to them. Let me just keep going through this, if it will let me. Interestingly, although we have a record of eukaryotes that goes back at least 1,600 million years, when we look at the biomarker record here from the work of Yuck and Brox, we find that there's not much record of eukaryotics in the form of lipid biomarkers. Indeed, again, from this middle Proterozoic basin, we have evidence that most of the primary production was bacterial, and indeed, in this particular basin at this particular time, the lower part of the photic zone was anoxic, because we see biomarkers that are made by green and purple sulfur bacteria. Now, although eukaryotes originate early, about 800 million years ago, we see a major diversification with new types of fossils like these vase-shaped tests. We start seeing scales of the type that some of you might associate with coccolithophorids today. We see an increase in the diversity of multicellular and cenocytic types. We see an increase in the diversity of cysts and other types. And one might ask, why halfway through the history of eukaryotes do we see this difference? And let me suggest an ecological argument, and you can take this for what it's worth, but many people think that there's at least an ecological component to the driving of diversification of animals in the Cambrian that is associated with carnivory. And so if there were a significant uptick in the ecological importance of protistine eukaryotes that eat other eukaryotes, that might drive much of what we see in the Neoproterozoic. And indeed, when you do a sort of character analysis with phylogenies, it's pretty clear that eukaryotes that make their living largely by eating other eukaryotes phagotrophically are derived groups whose molecular clock ages suggest an origin around, at least in the Neoproterozoic. And certainly fossils, these vase-shaped fossils, which are abundant in rocks beginning around 750 million years ago, clearly made by things like testate amoebae today. Some of their modern relatives are, in fact, eukaryote eaters, and one can guess, A, that the reason you have a test is for protection, and B, there's actually evidence of predation on these things. Also, we find, beginning in rocks of this age, that we start seeing scales. Indeed, these scales described by Phoebe Cohen are probably the most diverse eukaryotic fossils before animals. And again, while we can't pin them down phylogenetically, there is reason to think that they might serve the role of protection against other protists. And then finally, one can think of multicellularity as a defense. And just to argue this, one more point, then I'll finish. there's a wonderful paper some years ago in which people were growing single-celled algae in culture, and you can grow these for 1 million generations. They'll always be single-celled. You put in a eukaryotic predator, and within 10 to 20 generations, they're obligately multicellular. And they will stay multicellular even if you remove the predator. And the reason for that is that when you stay attached after cell division, you can't get eaten. So there may be something that, since molecular clocks suggest that animals are taking shape on this time scale, maybe that has something to do with it. Last point. There's a conundrum here in that we think that eukaryotes existed early. We have evidence that photosynthesis came to eukaryotes fairly early, yet if you look at the biomarker record, it looks like eukaryotes are only becoming important in the phytoplankton about 700 million years ago. And again, I think ecological modeling can help us. On the right is an interesting diagram, and what it shows is that if you look at basically population size, which is what we're really looking at on the x-axis, and just look at the, size is on the x-axis, and population numbers on the, body size on the left, on the y-axis, numbers on the right. At low nutrient levels, small cells always win. And so today, in the lowest nutrient levels of the ocean, cyanobacteria dominate primary production. And it may well be that the reason cyanobacteria dominate primary production through most of the Proterozoic is that nutrient levels are very low. Independently of any paleontological evidence, there's evidence for a change in phosphate availability beginning in the later Neoproterozoic. And what will happen ecologically if you add nutrients, you increase the population size of the cyanobacteria, but only to the point where they become controlled by grazers. And at that point, larger size classes can grow in. So it may be that the reason that eukaryotic algae become important only toward the end of the Proterozoic is that nutrient status is changing in the oceans. And that has a further consequence, because once you replace or add larger cells at the base of the food chain to the smaller ones, you end up distributing more energy and biomass higher in the food web. So it may be that at least one component of the riddle for why animals radiate when they do is that the changing nature of the primary producers in ecosystems is actually making more energy and biomass available for larger organisms at the top of food webs. So with that, then, let me just end. Punchlines: life and environment are recorded over a 4 billion year history of the Earth, and that they seem to be closely intertwined throughout that history. The Proterozoic is a world that is both biologically and environmentally distinct from what became before and after. And importantly, that Proterozoic rocks provide maybe our best template for thinking about how to interpret the deeper rock record, and also provide a guide for the exploration of sedimentary rocks on Mars and elsewhere. So thank you very much.
Thank you very much for a beautiful talk. Yes, please.
In the Phanerozoic, there are quite a few mass extinctions, and I'm wondering, during the 2,000 million years of the Proterozoic, whether are there any sediment layers that are well preserved enough and chronologically ordered that you can test or try to see if there were any mass extinctions?
Yeah, I guess I think I need to use this. There's at least one case we can make. People talked yesterday about these global glaciations, sometimes called Snowball Earths, one that begins actually rather precisely at 716 million years ago, ends about 660 million years ago, and then a second shorter lived one that ends 635 million years ago. If you look at the nature of the eukaryotic biota before the earlier of those ice ages, you have this relatively rich diversity of a variety of eukaryotic clades, very few of which actually make it through the glaciations. Now, we know that some eukaryotic clades did, because there's reason to believe that some of the diversity that radiates after the ice ages has its roots in this earlier diversification of eukaryotes. But at the species and genus level, not much makes it through those ice ages. So that's our one best look at mass extinction. Now, for prokaryotes, bacteria, it's a little bit different in that, as I always tell my students, you can brush your teeth in the morning and kill 99% of the bacteria in your mouth, but by evening, your teeth will be filmed again. So I think it's a different order of exercise to try and drive bacteria to extinction, and we certainly see no evidence of it.
So when people say that the Permian-Triassic extinction was the largest ever, that's probably not true because of the one you just described?
Well, you know, it's very hard to quantify what's going on. So I sometimes write about the Permo-Triassic, and I need to be able to say it's the largest mass extinction ever. So I'll stick to that story. The extent to which it's true, we simply don't know.
Any other questions?
How much of a shift from cyanobacteria to eukaryotic algae could be a result of increased availability of fixed nitrogen?
Well, I guess one of the questions is, it could be important, and Ariel Ambar and I wrote about this some years ago. In some ways, I think that nitrogen may prove to be a responder here. I mean, there is evidence based on nitrogen isotopes that at least in shallow water environments, there was some nitrate available. As you go offshore, nitrogen isotopes indicate a greater role for nitrogen fixation. And certainly, one of the things I didn't have time to talk about, but environmentally, in literally every Proterozoic basin but one, within the range of basin depths recorded in sedimentary basins, which is really only a couple hundred meters at best, you capture the transition from oxic to anoxic water masses. So most of the Proterozoic appears to be this fairly thin veneer, probably tens of meters of oxic waters beneath an anoxic oxygen minimum zone. And there's no question that's a perfect conditions for actually destroying nitrogen and leading it back to the environment. So yes, at low nitrogen, particularly offshore, one might favor cyanobacteria. One of the things that I think will build up nitrate availability in the oceans is simply as you create a more oxic redox profile. And that, I think, is ultimately driven by phosphorus. So yes, it's important, but yes, it's not independent of what's going on with phosphorus.
Do you see any changes in the physiology or the shapes of eukaryotic, I guess, protists at the GOE?
Well, we don't have any. The oldest fossil evidence that we have for eukaryotes is about between 1600 and 1650 million years ago. Now, having said that, one has to be honest and say that the quality of the record per se just gets worse and worse as you go backward through time. But no,
How about prokaryotes? Same question.
Oh, for prokaryotes, well, I think what happens that is very different is that to the extent that cyanobacteria existed in the Archean Earth, their population sizes and oxygen fluxes associated with those were insufficient to essentially titrate out all the sinks for oxygen. What happens at the GOE is that, again, because of oxygen produced by cyanobacteria, you end up oxygenating the surface ocean and atmosphere. And that shows, I think, an important thing, that cyanobacteria end up being ecosystem engineers, in that through that process associated with the oxygen they produce, you basically produce photic zones in which the alternative electron donors, things like sulfide and ferrous iron, are simply no longer there. So I think that's the big difference then is cyanobacteria either go from being absent or a smaller part of ecosystems to being dominant primary producers. And of course, we don't see this in the fossil record, but once you get metabolically significant amounts of oxygen in the atmosphere, then you dramatically expand respiratory capabilities, chemoautotrophic capabilities, biosynthetic capabilities. And I think you see some evidence of that in phylogenies, but not directly in the fossil record.
But as an extinction event, do you think it trumps the one that you just described?
Well,
a few minutes
Lynn
ago?
Margulis, bless her soul, always used to talk, I think unfortunately, about the oxygen holocaust. And she had this sense that basically the world was full of anaerobes, and as oxygen comes up, they all die. Actually, what they all did was went a millimeter beneath the surface, where there's still anoxic environments. So I tend to think of the GOE as expanding the ecological amplitude, not simply changing it in a way that fundamentally drives most earlier life out of existence.
I guess I basically agree to your scenario, and you have continuously written that kind of work. And I have read your papers from your rank stage. And my question is, I'm a geologist,
Yeah.
and we have worked over the world. It's more than 100 localities over 30 years. And then the Archean is a kind of no sedimentary rocks,
Yeah.
nearly absent.
Yeah.
So that therefore, in your opinion, okay, quite few amounts of nutrients provided.
Yes.
And the Phanerozoic is the emergence of huge landmass because large amounts of sedimentary rocks appeared. That's fine. And then probably the cyanobacteria produce oxygen, and oxygen pumps have been really productive.
Yep.
So that you mentioned, okay, it's surface high, from low oxygen, intermediate, and high oxygen.
Right.
So the there is the okay, then life change, very simple one prokaryote, right? And then endosymbiont.
Mm-hmm.
And then we are multiple architecture, including everything.
Right.
So that, okay, my question is what is the most important environmental pressure to change the system of life?
Okay, that's a very
Yeah.
good question. And
Yeah.
I think I moved over a slide from a review of Tim Lyons's papers quickly, but other people have shown similar things. That I think that most of us would agree that before the GOE, O2 tensions were very, very low, 10 to the minus 5 PAL or something like that, that you permanently have a transition to a world that at least has something like a few percent of today's oxygen levels in the Proterozoic, and that persists. It's usually shown as a straight line, but that's a line of ignorance. It could go up and down. We don't really know what's going on. But all the sedimentary geochemical indicators tell us that we are moving to a world with more completely oxic redox profiles about the time that animals come in. So yes, I think there is a history of increasing oxygen. There's a paper just came out by two geologists in the United States, John Husson and I'm just blanking on, Shannon Peters, in which they actually argue what may be similar to what you're thinking is that you simply have more erosion and more sediments in the Phanerozoic than you did earlier, and that's what drives this oxygen change. I think the problem is we know that there is a decay curve, that the older the sedimentary unit, the more likely it has been eroded. So I think that I would agree with previous speakers that you probably have less exposed land mass in at least much of the Archean, and that may change nutrient levels. I think the difference in preserved sediments between the Proterozoic and Phanerozoic may well be a question of preservation. So to get at your question then, what people have stopped doing is saying, why was there 1% of today's levels in the Proterozoic? Or they've stopped saying, why did oxygen change at these two junctures at the beginning and end of the Proterozoic, but rather say, what conditions would actually maintain a long-term steady state of these three different types? And again, that appears to go, perhaps more than anything else, to nutrient availability, especially phosphorus. And the important thing that's becoming clearer and clearer is that in a world where anoxic water masses are rich in ferrous iron, there are important sinks for phosphate that marine geochemists haven't thought about until recently. Soil chemists knew about these 30 years ago, but marine chemists are just starting to think about
sorry to interrupt, but sorry. My final important question is why life selected, the size, for example, 1 million bigger for eukarya, and another 1 million and two bigger, that is us, but multiple assemblage of many different organisms and
if I understand the question, then I
driving force.
Yeah, I think there are,
for example, metabolism.
There are diffusion plays a very important role on limiting the size of organisms, and I think bacteria are largely small because of diffusion and the nature of their cytosols. Eukaryotes, which have a different internal architecture, can be somewhat larger but are still diffusion limited. The nice thing about animals, at least once you start climbing the animal tree, is that they actually have structures that essentially get around the limits of diffusion. I mean, you circulate oxygen through your body. So I think in some ways it's then the relationship between the physiological capabilities of an organism that are related to size and essentially the amount of oxygen, which in turn actually does go back to things like phosphorus availability. So it's not at least I don't think there's a simple magic wand, but I think it's this interconnectedness that goes from really physiology to tectonics.
Yeah, thank you.
I'm sorry, but it's time. So, another question? Okay.
It's a related question to the phosphorus story you just suggested. So one implication of that is that continental weathering rates may have been constant for 1 billion years, which is interesting to think about.
Yeah, it really is. And again, when you look at the Tim Lyons curve, I mean, you showed something like it. It shows the atmosphere being, having a constant composition for 1 billion years. We don't know that that's true. All we know is that it never dipped below, back into atmospheric anoxia, and it never reached a state where you essentially eliminated all of the subsurface anoxic water masses. But that means it probably varies within a certain level. So yeah, it could be. I mean, what I don't understand, that the middle of the part of the Proterozoic, where the carbon isotopic curve is flat, which is odd, where you have, and you know more about this than I do, anorogenic granites, odd structures and petrology, sometimes called the boring billion. What's really going on? Fundamentally, I don't think I understand the tectonics of that portion of Earth history, but it does seem to be, at least qualitatively, somewhat distinct from the Phanerozoic tectonics that we understand and earlier stuff. That's the only thought I can give you.
Okay. So let's thank the speaker again. Thank you very much. Before closing the session, let me make an announcement for the poster presenters. please remove your posters during this next coffee break, so that we can clean that room. So let's thank the speaker again, and thank you very much for your active discussions.